yours.
Thank you very much, Fernando. Well, it's a pleasure to be here and to present this talk. Well, the first thing you wonder probably is why this title. Well, even we publish in Genomics, and the talk today is going to be about genomics. Our group try to be a plant pathology group, and this is something that is obvious for somebody, but it's not so obvious, especially for a student when he came to the lab and they want to do molecular biology, they want to do genomics, but we are plant pathologists. We use genomic or use molecular biology as a tool, and even we are doing some basic research. Our goal is not the basic research because. Okay. Okay. Even we are doing this genomics or genomics analysis. Our goal is not this for basic research or to publish this because we wanted to help the agriculture, and we wanted to what we want to do is to solve problem for them. And I try to convince during today that what we do the genomics and why we use this tool for plant pathology and to solve problem for agriculture or and the producers. This is a summary of what we're going to talk today. I will give you a brief introduction on two diseases we were working on. One was in US when I was working in US, and the other is Xanthomonas citri pathovar citri. That is you will see that it's a really important disease as a quarantine pathogen in European Union, and we were working in Florida for a while. And we are working also mainly in Xanthomonas arboricola pathovar pruni that used to be also a quarantine pathogen. It's not anymore. Now it's a regulated pathogen. I will talk to you later. And in both cases, we have some problems with misidentification of the strains of the bacteria that cause the diseases. You will see. And later, I will talk about the past, something that we could solve using genomics, and we didn't because we have not the tools at this time. We talk about Xanthomonas arboricola pathovar pruni. In this case, we have the tools, and we could solve some problems for agriculture in Spain. And we talk a little bit about the genomics, about the analysis in Xanthomonas arboricola for bacterial identification, but also to discover factors involved in pathogenicity. Well, probably Fernando talked to you about this sometime. Xanthomonas is a really interesting group for microbiologists and also for plant pathologists. Why? Because it used to be defined as a plant pathogenic bacteria, but this is not really true now. I mean, now we know that it's not only a plant pathogen, it's also an endophytic bacteria that is in most of the plants right now. Well, but it's really interesting because we have many species, many pathovar, and the good thing for us and the interesting thing for us is that it's affecting specifically some host. I mean, one species on one pathovar is affecting one species and is not affecting the other. Of course, they have different infection, colonization process, and disease diversity. And today we are going to talk about, as I told you, Xanthomonas citri subspecies citri and Xanthomonas arboricola pathovar pruni. If you see different names, it's because one of the things in Xanthomonas is the taxonomy is crazy. The taxonomy in Xanthomonas is crazy. The taxonomy is changing almost every month. I mean, and you will see different name for the same for the same species or for the same even for the same pathovar. But as I told you, it used to be defined as a plant pathogenic bacteria group, and now we know that it's not really true. But even in this host, in citrus or in prunus species, we have non-pathogenic bacteria, and this is something that is really problematic sometimes, especially for regulators and for people that for plant protection service in everywhere in Spain, especially in Spain, they have many problems with this because they found some of these Xanthomonas and they thought or they think sometimes that they are pathogenic and they are not really pathogenic and must not be regulated. Well, this is something that Fernando knows very well with the Juglandis, and they did a really good work defining a new species. Going to the past, well, this is Xanthomonas causing disease in citrus. In citrus, there are two main Xanthomonas. One group of Xanthomonas that's called citrus canker, and citrus canker is a quarantine disease. It's one of these diseases that is called devastating disease, and it really, if you have citrus canker, you are losing a lot of money, and it's economically is really, really important. Well, in Florida, this is in Florida. In Florida, they have also they have Xanthomonas citrus subspecies citrus. I remind you that it's not in the European Union, and but they have another Xanthomonas that is called the disease is called citrus bacterial spot. This is not an important disease. It's affecting young plants. The young plants, you have some spot on the leaves. Actually, with copper treatments, you can solve the problem. And even without any treatments, sometimes the adult plants are free of the bacteria. What happened in the past? Well, in 1984, and this disease was found in Florida, and they called citrus bacterial spot, and was at this time the bacteria was called Xanthomonas campus pathovar citrumelo. And the disease appeared. The symptom of the disease was flat lesion with necrotic center on water socket. So water socket is typical on Xanthomonas. And initially in this year, this bacteria was considered a new form of citrus canker. And this is a really important issue in Florida at this time, because was considered citrus canker, and at this time, the citrus canker was under eradication in Florida. And this means that all areas with this bacteria, in all these areas, trees were removed and nurseries were closed. Well, was completely devastating, even more than citrus canker, the real citrus canker. But well, at this time, over 20 million trees in more than 100 nurseries and orchards were destroyed, and this cost about $100 million. Many nurseries were subjected to lengthy quarantines. Many of them were closed. I mean, was a real problem for just not a social problem, a political problem, an economic problem. But in September 1990, all the regulation for this strain were removed, because during these 5 years, researchers realized that this bacteria was not causing a real problem in the trees, in citrus trees. All of this was necessary. Well, this is the question. Now it's easy to answer the question. Was not necessary. I mean, during 5 years, they were removing trees that were not causing any problems and will not cause any economic problem in Florida. But at this time, they didn't know what happened. This could be solved if in 1 week or less than 1 week we had the genome, because it's really easy to distinguish or to discriminate between citrus canker strain and this strain that are almost no virulent. Well, this is a slide I used to put in Florida because I was working with both models in Florida, with citrus bacterial spot and citrus canker. And I used to put this for a student, mainly for a student, because in Florida they have also panthers and they have cats. If you see, this is the same genus but different species. And it's really important. If you have a panther in your garden, it's going to be a problem for you. Not because the panther is not doing anything most of the time, it's because you have to call the animal protection service and they have to remove the panther from your garden. If you have the cat, probably if you like the cat, you are going to keep the cat at home, no problem. This happened also with these two bacteria. At this time, now it's not citrus canker is not eradicated in Florida. They have other problems and they are not eradicating citrus canker. But if at this time, if they had citrus canker, they have to call the plant protection service, and they were going to remove your tree, not just your tree, but all the citrus around. If you have a citrus spot, a citrus bacterial spot, there is no problem. But the problem for this is how you identify one and the other. And we solve this problem with genomics, but not NGS, because at this time it was not so easy to obtain the genome of both strains. And going to the right part of the slides, well, this is Sun Tzu. Sun Tzu say that if you know the enemy, you are going not you don't need to fear about the result of 100 battles. This is like this. If you know you have this or you have this, maybe you don't need to do anything if you have this, and of course you need to do something if you have here. The knowledge of the content means better knowledge of the microorganism in case of plant pathology, and this can be solved with the genomics and the next generation sequencing. And this is what we are doing now, next generation sequencing for this type of problems. This going to decide, NGS also allow us just not just to know if you have the pathogen or you don't have the pathogen, is to know what you have in a plant. If you can now, if you have all the microorganisms you have, all the virus, fungi, or bacteria you have in a sample. I'm not going to talk too much about this, but this is also you need to consider if you need this in a sample. I mean, because now some of the people are saying that, well, in a future, in a plant protection service, you are going to analyze everything you have in a plant, and this is sometimes problematic, because you can and it's happening now. I mean, if you analyze, for instance, a grapevine or a vineyard, you are going to find many viruses in the vineyard, but it really needed to know what you have, all you have there, because sometimes it's a problem. Because if you are going to tell the people, you have these 100 viruses, you need to know if these 100 viruses, or at least some of these viruses, are doing something in the plant. Take care about this, and regulators must consider this. I mean, you don't need, or you need to think about what you have in the plant, and if you, in the plant, the microorganisms you have in the plant are doing something, or are causing any disease, because not just plants, but also humans, animals, we have microorganisms that they're not doing anything. I mean, are symbiotic, and they're not doing anything, and this is kind of now is something that we need to think about it. Well, but no, I'm not going to talk too much about this. But genomics and next generation sequencing allow us to identify causal agents, disease causal agents, and also understand the genomic mechanisms that underlie the disease. And this is what we are doing now with our bacteria in our group. Well, this is the model we are mainly working in our group now in Spain. Is Xanthomonas arboricola pathovar pruning. We are doing some identification, and we are studying some virulence factors. What is Xanthomonas arboricola pathovar pruning? Xanthomonas is causing a bacterial spot of stone fruitan, especially for Spain is really important almond. Well, it's a gram negative, allowing to that allow to Xanthomonas arboricola species. Now it's in the EPPO A2 list, but I think 2 or 3 years ago was still a quarantine pathogen in European Union. Now it's a regulated non-quarantine pest, and this means something, that the plants in nurseries or plant producer must consider this disease and cannot sell plants with the bacteria. Well, host plant is Prunus spp, and it's affecting apricot, plum, peach, Japanese plant, and I put here cherry, but you will see what happened with cherry. I will show you some of the results we have on cherry. It's a serious disease because it's difficult to control, easy dispersion, and cause high economic losses. And one thing that is really important is with Xanthomonas arboricola pathovar pruning strain, we have also some other Xanthomonas in prunus that are not doing anything, and used to be classified as Xanthomonas arboricola pathovar pruning. You will see. Well, Xanthomonas arboricola pathovar pruni, these are the symptoms. You have this spot on leaves, and this is important also symptoms in, or mainly in fruits. And for us, for Spain, it's really important symptoms on the on almonds, because we are a high producer, almond producer. Well, the disease is all around the world. Most is present right now in most of the areas where the prunus are. And in Spain, you will see that we have the map, and almost in everywhere in Spain, you can find the bacteria now. When we start to work with Xanthomonas arboricola pathovar pruni in Spain, first thing we did was to collect a collection of a strain from the plant protection services. And these strain were classified as SAP, as Xanthomonas arboricola pathovar pruni, because they were identified by a PCR that was based on an ABC transporter in the bacteria. What happened that when we start to do the analysis, deeper analysis on the strain by multilocus sequencing analysis, we realized that, well, we have some of the Xanthomonas arboricola pathovar pruni that were close to reference strains, but we found some of them that were not close to this reference strain, were out of this group. And this I mark here and in black, and we have different, at least 3 different strains at the very beginning, but just 3 at the very beginning, but later we found much more. And these strains were positive with these ABC primers that they identify all the Xanthomonas arboricola pathovar pruni. Was a big problem for producers, because most of these strains were found in nurseries, in plant producers, and this was well, this was part of a Garita Cambronero PhD thesis, and what we do, what we did at this time was just to identify. I mean, by multilocus sequencing analysis, we knew that we're not really Xanthomonas arboricola pathovar pruni, at least in genomics, based on genetic features. We realized that we're not Xanthomonas arboricola pathovar pruni. We call these strains Xanthomonas arboricola pathovar pruni lookalike strains. First, of course, we try to find if this strain causes any symptoms in prunus, and we realized that none of them produce any symptoms on prunus. Well, I don't see, you cannot see here, but well, this is one of the strains, and this is probably the control. This is Xanthomonas arboricola pathovar pruni in prunus, and this is one of these lookalike strain. They didn't cause any symptoms in prunus. We did this also biochemical analysis. At this time, we were also interested to check other characteristics like motility and, well, different type of motility, and were completely different. These strains were completely different to the SAP strains. What we did also was to inoculate the strain in several other hosts, and we found no host for this. We didn't find any host for this strain. Just one of these, this one that was pathogenic in some host, but was not really a prunus species. And at this time, we did, okay, now we need to know what happened, which is the difference between those pathogenic in prunus and those that are not pathogenic in prunus. And of course, we obtain the whole genome. And the whole genome confirmed what we have on multilocus sequencing analysis. We have some of the strains that were not considered or could not be considered under a genomic perspective, Xanthomonas arboricola pathovar pruni. Well, here you have a substrain, and here you have this lookalike strain. And you see that there is a variability among Xanthomonas arboricola pathovar pruni lookalike strain. I mean, not all of them are the same. Well, at this time, we realized that we have some of them that are Xanthomonas arboricola, some were not Xanthomonas arboricola, and the genomic content was different among them. Well, this is just to show that, of course, most of them are Xanthomonas arboricola are share most of the genes. I mean, because they are very close. You should consider, or you must consider, that are the same species but different pathovar. I mean, the genome is quite similar among all the strains. But for us was really interesting because it's similar to what Fernando's group is doing. I mean, it's different Xanthomonas in the same host, and the behavior of them are completely different. And this is the hypothesis. The first hypothesis was, okay, this is because they have some different elements in the genome that confer this characteristic in the different behavior. I remind you that most of them are not pathogenic, and none of them were pathogenic on prunus. Well, at this time, as I told you, I have a student that was working with this genomic analysis, and I told, okay, now we need to know why one of the strains are pathogenic and the other strains are not pathogenic. And we start to analyze different things, the different secretion systems in the bacteria. I will explain later more about these secretion systems. And we analyze several things. We analyze, for instance, MCPs that are receptors for chemotaxis, some adenosine. We found some difference in the flagellin and in the different transporter, and well, we realized that these lookalike strains were different, and we found a specific difference that maybe contribute to the. At this time, we thought that maybe they are contributing to the different virulence of the different strains, or the different type of strains. Well, this is type 2 secretion system. I'm not gonna go in deep in all the system because we have no time. But this is type 2 secretion system that is secreting peptolytic enzymes, cellulitic enzymes, it's different enzymes. And this is interesting because, and this is one thing that now we have confinement with other non-pathogenic strain, and it's interesting because we have a difference in these enzymes, and this is probably because the different lifestyle. Probably the non-pathogenic strain need more of this than the pathogenic strain, because the pathogenic strain are able to release compounds for the life through the pathogenic, the virulence system. This strain, non-pathogenic strain, looks like they need more of these enzymes and have more genes for this enzyme and secreting more of these enzymes. But for us, the most important thing was to analyze the type 3 secretion system. Type 3 secretion system is the one that is described to confer pathogenicity or virulence in most of the plant pathogenic bacteria. And of course, we realized that this is similar to Fernando's work, that we found some of the strains that have the type 3 secretion system. Some of them, they don't have the type 3 secretion system. And these have the type 3 secretion system, but it's not I mean, the type 3 secretion system is the same, but they have just a few of the effectors. I mean, for this strain, you have about 20, 21, 22, 23, depending on the author, effectors, and this 22 or 23 effector confer the virulence to the bacteria. And in this one, we have just two, or the 2 from up to 6 effectors. And I remind you that this strain is able to produce symptoms in some herbaceous species. Well, the important thing is that well, there is a huge difference in the effectors. And as I told you, I mean, this is nice work because at this time you can hypothesize that okay, these effectors are the difference between or among strain, pathogenic and non pathogenic strain are based on the type 3 secretion system. We will talk later about this, but the most important thing here is that we use one of these effectors to design a diagnostic protocol for Xanthomonas arboricola pathovar pruni. I mean, because we know that some of these effectors are specific for Xanthomonas arboricola pathovar pruni. The first thing, as I told you, we need to do apply research, and we need to solve problems. We design a PCR protocol to detect the bacteria, specifically detect the bacteria that these Xanthomonas that pathogenic on prunus. And this is what we did. Well, this is an scheme of what we did, and we developed this PCR protocol that is based on this difference between, well, this you don't see here too much, or I cannot see at least. These are this one with a few effectors. These are the two arboricolas pathovar pruni, and the other one are those that are not pathovar pruni. And one of these effectors, this I think is this one, is not in any of the strain that are not pathogenic in prunus. And this effector now was used to develop this PCR protocol, and actually this is an official protocol that is now proposed by the EPPO, that is the official regulator of, or at least the one that proposed the official protocol for the European Union. Well, that's just well, this is what we did. I mean, this is the SOPI3 that is an effector that is specific for Xanthomonas arboricola pathovar pruni. When I say that is specific for Xanthomonas arboricola pathovar pruni, I'm not saying the whole truth, because, well, this is the difference between SOPI3, that was the gene we use for designing the primer and to propose the new protocol. And this is the ABC transporter that is, well, you see, you have this ABC is present in most of the Xanthomonas. I mean, it's not really specific. But I told you that it's a specific protocol, but I'm not telling the whole truth, because this SOPI3 can be found also, it's not found in any of the sub lookalike, it's not found in any of the substrains that are in prunus, but you can, this is a common effector in other Xanthomonas, and for instance, it's present in Xanthomonas citri, subspecies citri. But the thing is, you are not going to find this citri in prunus because it's specific for citrus. I mean, you find this OP3 in prunus is because you have substrain. I mean, it's easy to find difference between among the strain with the genomics, but you need to go further, because maybe sometimes you're gonna find some other strain with SOPI3 in prunus. But this, I tell you, even if we design the protocol, you cannot say that it's 100% specific, because you find this effector in other Xanthomonas, also pathogenic Xanthomonas in other hosts, but other Xanthomonas. Well, when we did the first work, we did this analysis with this genomic with just Illumina genomes, and was okay. For us was enough. At this time was enough. But if you want to have better genomes, you have to do something like this. It's hybrid genomes. This means that you sequence by long reads and also short reads, and you have hybrid assemblies. Actually, we are not doing hybrid assemblies. What we do is assembly by bio, the long reads, and correct with Illumina Novasec. And now we are thinking that maybe we are doing too much, because many times we don't need this Illumina. We are correcting because with PubBio now we can have closed genomes, and was just one contact for the chromosome and one contig for the plasmid. Well, we did this because now we try to go further, and okay, now we know the difference between Xanthomonas arboricola, pathovar, pruni, and they look like a strain that are in prunus. But going further, why Xanthomonas arboricola, pathovar, pruni is infecting prunus, and why Uglandis or Uglandis and corylina are infecting walnut or halzenut. Well, this is what the first goal of this work. And again, well, I'm not going to talk too much. We have some markers now for each of the pathovars, of course, with these high quality genomes. But the important thing here is also that I'm not going to talk about this whole range of factors, but we found some difference between the different pathovar, and this factor for the different factor for the different pathovar also allow us to design a specific PCR protocol for each one of the pathovars, because we are comparing very close strains inside the same species, but with difference in pathogenicity or in virulence. And this is one example of what this is just one, but we have now about 8 or 10 different protocols that are able to discriminate between the different pathovars. What we did is, for instance, compare TBDTs that are TOMB transporters, and we found difference in the TOMB transporter among the different pathovars. We do blasts, and we found variability regions, and based on these variability regions, we are able to design a specific protocol for each one of the pathovars. And this is one case that I told you at the very beginning. I mean, when we talk about Xanthomonas arboricola, pathovar, pruni, we used to say that affect apricot, peach, cherry, plum, and cherry. And we were really interested in cherry strain. And during the year, we were looking for cherry strain because it's described in different groups around the world, some people in the US, some people in Italy. But we couldn't find any strain from cherry because these people, some of them told us that they have no the strain, they lost the strain just a few years ago. The other didn't reply to us, and we didn't have any cherry strain, and we're really interested in cherry strain. What happened 2 or 3 years ago, one group in Montenegro published a paper describing the Xanthomonas arboricola, pathovar, pruni in Montenegro for the first time. This was a first report of Xanthomonas arboricola, pathovar, pruni in Montenegro. And among the hosts, they describe some strain in cherry. Okay, we asked these people in Montenegro, and okay, we established a collaboration, and we have sequenced this strain the same with PacBio and Illumina. And we obtained 2 different genomes, and we analyzed this for everything. I mean, in this case, we did the whole work. I mean, we analyzed type C secretion system, factor involved in virulence, and everything. And we realized that what they identify in cherry are not really pathovar, pruni. Actually, it's something different that we were not able to demonstrate that these strains are pathogenic because we inoculate in cherry are not giving any symptoms. They say that, but it's maybe because the different cultivars, the cherry they have in Montenegro. But here in Spain, it was impossible. I mean, we didn't reproduce any symptoms. And we analyzed, of course, by multi-locus sequencing analysis, and also the type 3 secretion system. And it's clear that the bacteria, this strain, has the type 3 secretion system, but has not the same effector than pathovar, pruni. This probably is happening what happened with your strain of Xanthea, because the type 3 secretion system is there, they have about these six effectors. This means that they may produce some symptoms in some hosts, but this cannot be considered Xanthomonas arboricola pathovar pruni. And this is going to be published probably in a few weeks. And also, if you do an ANI or FASANI or PAIANI or whatever you want, this strain are classified as not Xanthomonas arboricola pathovar pruni. This is really interesting because now we wonder if this cherry strain really exist. I mean, because we don't have any strain from cherry that are considered were identified as Xanthomonas arboricola pathovar pruni, but maybe it's happening the same than Neurosantea. You identify some Xanthomonas in Juglandis and think that you have this Xanthomonas arboricola pathovar Juglandis, but are not really Juglandis, and you have clear result. This is quite similar. This is interesting, and we're gonna go ahead to check why they are pathogenic in cherry, if they are. Well, of course, we inoculate the strains in cherry, and we analyze, compare with this really sub-strain. This sub-strain, you have symptoms, and this you don't have any symptoms in other prunus. But even in cherry, what you have is like a hypersensitive response, and we know that this a hypersensitive response, and it's HR, because the bacterial population is not increasing during the time. I mean, when you inoculate, you have at the very beginning you have a symptom, but during the time course, the population is decreasing. This means that you have no really an infection. What you have is something similar to an HR, and this. We don't know yet, but this probably what they have in Montenegro. As I told you, this is going to be published in a few with the people in Montenegro, of course, and it's going to be published. But what you see is similar to the problems with the negative control, with a negative control. Until now, well, I show you some of the results we have that help in case of diagnosis in the cherry strain or some lookalike strain. But all the genomics also allow us to go deeper in the plant microbe interaction. And we are really interested in type 3 secretion system and type 4 secretion system. I will explain why. Well, this is the same because most of the work was done with this in genomes, with this Bio and Illumina Novasec, because we were thinking that to do this kind of work, we need to go to have these high quality genomes. Sometimes I tell you that you don't need this high quality genome, because some of the results we have here are exactly the same that we have just with Illumina. I mean, with different contexts. Here, the thing is we have one context for the chromosome and one context for the plasmid. But actually, right now, we are analyzing more than these 6 strains. We are analyzing about 100 strains in another different project, because we are comparing virulence of these strains with tomatoes and pepper strains, and we don't need these high quality genomes to find what we found here. Well, these are secretion system in the bacteria. Probably you are familiarized with all of this, and we are studying type 3 secretion system because we want to analyze the virulence and pathogenicity of the strain. But right now, we are also very interested to find or to study the fitness of the bacteria in the plant, inside the plant. And especially, we are really interested to find factors that affect the competition of the bacteria to survive the competition with other bacteria. And in this case, we are studying the type 4 secretion system and type C secretion system. We are not doing anything because we don't find this type C secretion system. And we are doing this, in this case, we are going ahead, and we are studying not just the genomics but also functional analysis of the elements that differentiate one strain and the other. Well, the first is when we analyze the whole genome, and this PacBio Illumina combination genomes, we realize that we have a specific region that is repeated in the plasmid and in the chromosome. And this was done by a student that was doing an stay in Montpellier with one guy, Ralph Koenig, in Ralph's lab. And Ralph told us, okay, you have a problem with assembly, and you have an error or a mistake in your assembly. And well, because Ralph told us that, we should, okay. At the beginning we thought, okay, this Ralph is like the god of the Xanthomonas, and we thought, okay, we're going to assembly again, and we assembly by different type of assembly, and we find exactly the same region here and here. Okay, and we thought, okay, we're going to analyze this why this region is repeated, because at this time, well, we thought, okay, maybe with different type of assemblies, we are having the same error. Well, but what we found was really interesting, because this region is a transposon derivative element. A transposon derivative element with some of the type 3 secretion system effectors. This means, or this makes you to think that may be important for, or really important, or the main element for the virulence of the bacteria, because it's a repetitive element, it's a transposon, and supposed to be in the chromosome, because the bacteria must to keep it to maintain the virulence. That supposed to confer an advantage on survival, bacterial survival in the plant. Well, of course, because Ralph told that was a mistake, we did everything to demonstrate him that was not a mistake. And of course, we did different PCRs in the chromosome and the and the plasmid to prove that it was really, and that's it. And well, that's it in some of the strain, but in some other strain, we have just the partial element in the chromosome, this transposon element in the chromosome. But when we went to the database, we found the same. Some of the strains of Xanthomonas arboricola pathovar pruni have this complete element in the chromosome, and some of them just partial element. I mean, it was confirmed that this transposon element was in all the strains, in all the high quality genomes in the database. And what we did, or what we are doing now, because we didn't finish yet we did some mutants. Now we have some mutants on some of the elements of the transposon. I'm not going to tell you much more about this because we need to finish, but the first thing and important thing is, until now we couldn't demonstrate the SOP3, the effector SOP3 is the main element, because when we mute the SOP3, the bacteria is able to reproduce symptoms in plant. This means that the first idea that the SOP3 was the main element of the virulence of this Xanthomonas arboricola pathovar pruni looks like not true. I mean, we need to finish this. We are analyzing other elements on the transposon, on this transposon, but is the other factors in the transposon element, and hopefully we will find which one of these elements is the main factor of the virus. And just to finish this talk, one of the things we are really interested now is in some of these type 4 secretion system that is present. By Genomics, we found this type 4 secretion system in all these Xanthomonas arboricola pathovar pruni, Xanthomonas arboricola pathovar juglandis, and Xanthomonas arboricola pathovar corylina. And we wanted this to demonstrate if this type 4 secretion system is doing something in the bacteria and is conferring some advantage over other bacteria in the microbiome of the plant. And what we did was to mutate this type 4 secretion system in Xanthomonas arboricola pathovar pruni, Xanthomonas arboricola pathovar juglandis, Xanthomonas arboricola pathovar cordiana. And yes, this element is working to confer the bacteria competition ability against microbiota in the plant. And this is proved by with first assay where is a model with E. coli. But we introduce in this in our assays also this Xanthomonas 44, that is a Xanthomonas arboricola look-alike. One of these strains we isolated at the very beginning that has not the type 3 secretion system. And we found also some competition conferred by the type 4 secretion system with Xanthomonas arboricola pathovar pruni against this 44. And it's really interesting that this also some strain of Xanthomonas arboricola pathovar juglandis even more ability to compete with against this 44, because for instance in this one, if knock out this BD4, that is type 4 secretion system, you will see that the mutant is not able to compete, and when you have the system, the bacteria almost kill the 44. But this is a competition assays that are addressed to know if these strains are able to compete with 44. Why we thought, okay, and what if 44 is able to compete with Xanthomonas arboricola pathovar pruni? Because from the very beginning, we thought that 44 could be used as a biological control agent, because if you mix in the same plant, we don't know yet, but in a first hypothesis, you can think, okay, you have an endophytic bacteria that is able to grow in the plant and is not doing anything on, is not producing any symptom. Okay, if you inoculate on the plant and compete with the pathogenic strain, can be used as a biological control agent. And we did this competition assays, the opposite. Well, this 44 is not able to produce any symptoms, but and is able to compete with one of the strain, at least one of the strain. Okay, I show you one of the strain of Xanthomonas arboricola pathovar pruni. But what we demonstrate here is that this competition is not based on type 4 secretion system, because this type 4 secretion system is not in 44. I mean, there is another way to compete with the pathovar pruni strains. And now we are looking which element of the 44 genome is able to confer this characteristic to the strain. But it's promising, because this strain could be a very good candidate to be used as a control agent, because it was isolated in Spain, was isolated from prunus, and would not so difficult to register as biological control agent. But we need to check in plant. And that's the final, just you to show some conclusion and reflection and takeaways. Massive sequencing methods are nowadays valuable tools, not only for research studies, but I hope I demonstrate to you that it's also useful for applied plant pathology. Of course, you need to take in consideration that these technologies may occasionally generate some problems. And I told you about for instance, about the microbiota analysis. That is really is something that I'm really worried about. Comparative genomics analysis through NGS has opened a world of possibilities for understanding the mechanisms that govern the infection, or to discover the keys of virulence of the pathogenic bacteria. And of course, I always say to my students, the study of bacterial genomes, in any case, should be complemented with phenotypic analysis that confirm or refute the hypothesis raised by genomics, because most of the mechanism virulence, or many of the mechanism virulence, are not because one gene or the other is present in the genome, is because of regulation. And we are doing also some transcriptomic analysis to go in further and try go deeper on this kind of on this analysis. And as final and obvious point, there is still much to be done to unravel the mechanism of infection and whole range of Xanthomonas arboricola. And Xanthomonas arboricola, as I told you, is a really interesting model because you have close taxonomic bacteria doing different things in different hosts. And this is for a microbiologist is really interesting. And that's it. Well, these are part of my group in Spain. Well, now we are involved in a project with not just Xanthomonas arboricola pathovar pruni, but also Xanthomonas in tomato and pepper, and with people of these different institutions, and also with the collaboration of companies, and Ralph is always behind this. Thank you very much.